the different stages and processes and parts of mitosis in the cell cycle. So here is a kind of a diagram of the cell cycle. We've been talking about this now for a few lessons, so we should, hopefully you're starting to kind of understand the general concepts. But after the cell's life officially begins after cytokinesis, you have like a growth phase where the organelles grow and the cell grows. This is called G1. This is the beginning of interphase. You can see here is interphase. And it goes to the S phase of interphase. This is where DNA is replicated. It's the new DNA is created. In human cells, we go from having 46 chromosomes to, during G phase and S phase, that number doubles to 92. Remember, we have two sets of chromosomes in our cells, each one of our cells, one set for mom, one set for dad. There are 23 chromosomes each, but so each cell has 46 chromosomes. So during interphase, it doubles up to 92, and that duplication completes. It's in a form called chromatin during this time. You don't see the chromosomes, the DNA is loose. G2, we go into another, it's a preparation for mitosis, where the cell goes through the different phases of division. Prophase, you begin to see the chromosomes form inside the nucleus. You have the centrioles, the spindles from the centrioles begin to kind of emerge during prophase. And then this is kind of like, this is still a part of prophase here. It's prometaphase, but the spindles come out from the centrioles and they latch on to the chromosomes. Metaphase, those chromosomes are lined up in the center of the cell. The way to remember it, M for metaphase means middle. The chromosomes are split. So you have, say for humans, you'll have 46 chromosomes going back to one side of the cell. This is during anaphase, another 46 chromosomes going back to the other side of the cell. In telophase, the two nucleuses, the nuclear envelope begins to develop. You have two distinct nucleuses begin to develop. You have the organelles, the two new sets of organelles are there. And in part of telophase, you have cytokinesis so for animal cells, you have two cells separate from each other. In plant cells, a cell plate would evolve, or develops, which eventually turns into a cell wall. Plant cells gaining their structure from the cell wall, so it's a little different for plant cells and animal cells, but they typically have, in eukaryotic, they have all the same organelles. Remember, eukaryotic cells, this process is for eukaryotic. And it's the division for prokaryotic is different than eukaryotic cell division. So that those are the different phases of cell division that are important for us to know. Let's go back up to this slide here. this piece right here, again, this is during S phase when and then again, they wouldn't see chromosomes like this in S phase. You would just have the chromatin. But these chromosomes here will appear during prophase, very beginning of mitosis. Another term we haven't really talked much about is homologous chromosomes, which all that means is that you have, it's a copy. You have two sets of chromosomes, so one from mom, one from dad. It's another way it's referred to as a homologous chromosome, and that is in each one of our cells we have a homologous set of chromosomes. It just means they're a pair of that information, DNA, that tells the cell how to duplicate itself and to do different processes in the body. But another term here is the centromere. That connects these two chromatids, which are just, again, chromatid is just another word for saying chromosome, but it's just a copy of a chromosome. And they're called chromatids. And again, they split during metaphase. After metaphase, when they line up in the center of the cell, they line up in the center of the cell during metaphase, and the chromatids split and they become chromosomes, and they head back towards each side of the cell, connected, with these spindles, guiding them back to their new sides of the cell, from again, the spindles coming from the centrioles. I'm kind of going over some of these terms again. These centrioles are what produces the spindles and allows the chromosomes to be guided back to each side. New organelles here, they represent the endoplasmic reticulum. You have the mitochondria. You have the lysosomes. You have all these different organelles they're developing on either side of the new cells. Cytokinesis occurs, and the cells start into the G1 phase of interphase, and so the whole process starts over again. So these are different processes of mitosis and the cell cycle that you need to know.